we should be able to get started. Oh, wow, it's already 9:21 here. Okay, we are on.
Okay.
The stage is yours.
Sure. A very warm welcome to everyone to today's presentation by our team, the Meliorists. Myself, Srushti Chipte, and my teammate Hemant Gabne will be presenting a paper called The Mitochondria Define Intestinal Stem Cell Differentiation Downstream of a FOXO-Notch Axis. To our non-scientific audience present, this will seem like too much to take in, but let's not gulp it all at once. Let's break it down to pieces first. So here are some terms and concepts that we need to get acquainted with before we delve into the presentation. First comes the phosphoproteins. You know them from our last presentation because they play an important role in the cell cycle arrest, DNA repair, and apoptosis. The phoxo family is thought to coordinate the balance between longevity and tumor suppression. As you can see, it plays a dynamic role. Next comes the biological pathways. A biological pathway is a series of actions among molecules in a cell that leads to a certain product or change in that cell. The two biological pathways that we will be frequently dealing with in this presentation are the WNT and the Notch signaling pathways. Again, this looks like a lot on plate, but right now all we need to know about these two pathways is WNT signaling pathway is a group of signal transduction pathways beginning with proteins that pass signal into cell through cell surface receptors. It has functions in carcinogenesis, that is cancer formation, and embryonic development. Also controls tissue regeneration in adult bone marrow, skin, and intestine. The Notch signaling pathway is a highly conserved cell signaling system, promotes proliferative signaling during neurogenesis, and plays a major role in regulation of embryonic development. Then comes the stem cells. Stem cells provide new cells for the body as it grows and replace specialized cells that are damaged or lost. They have two unique properties that enable them to do this. They can divide over and over again to produce new cells, and as they divide they can change into other types of cell that make up the body. Here's a representation of the adult blood stem cells. As you can see, they are located in the bone marrow, and later on they differentiate into different types of blood cells. In this presentation again, we'll be dealing with the stem cells of our intestine. Now comes the crypt-based columnar cells. Well, LGR5 is a receptor protein embedded in cell membrane that recognizes and binds to specific substances outside of the cell and is expressed across a diverse range of tissues such as muscle, placenta, spinal cord, and brain, and particularly acts as a biomarker of adult stem cells in certain tissues. Now, to visualize what are CBCs and where they're located, I've attached a short YouTube video. So let's see that first. The lining of the small. I hope this is audible and visible.
It's visible.
Yes. Intestine, like the lining of most of the gut, is a single layered epithelium. Depending upon location in the gut, the epithelial cells, usually called enterocytes, help absorb nutrients from the lumen of the gut or absorb water from the. The surface of the lining of the gut is increased enormously by thousands of villi that protrude into the lumen. Here we see a single villus and its internal architecture. The surface of the villus is covered by a single layer of enterocytes, which extend down into the crypt below. Stem cells at the bottom of the crypt, located between Paneth cells, divide and make copies of themselves, and also make transit amplifying cells.
The transit amplifying cells proliferate rapidly and move up the walls of the crypt. As the cells migrate upwards, they begin to differentiate into goblet cells and enterocytes. While
I hope the PPT is visible again. Okay, so am I audible?
Yes, you are.
Yes.
Thank you. So as we saw in the video, this is the villus, the crypt, and the stem cells, the stem cell niche. And here's a more explanatory picture for the same. Now, the intestinal epithelium consists of multiple cell types that derive from one single precursor, the LGR5 plus intestinal stem cell or crypt-based columnar cell. WNT and Notch signaling pathways are the main regulators of CBC's maintenance, proliferation, and differentiations. CBCs divide daily, producing rapidly proliferating daughter cells known as transit amplifying cells or the TA cells. Upon migration towards the intestinal lumen, progenitor cells become post-mitotic and differentiate into two main lineages, as you can see right here. This is the LGR5 plus cell, and it divides into two lineages: the absorptive lineages, which consist of the absorptive enterocyte, and the secretory lineage, which consists of the enteroendocrine cell, the Tufts cell, the goblet cells, and the Pennant cells. Now, Notch signaling is the key in determining the choice between absorptive versus secretory lineages. In fact, it acts as a node. While absorptive lineages sustain active Notch signaling, inactivation of Notch is requisite for secretory cell differentiation. In simpler words, when Notch is active, progenitor cells differentiate into absorptive lineages, and when it is inactive, they differentiate into secretory lineages. Further, it is shown that intestinal crypt CBCs and their adjacent Pennant cells are metabolically different. While CBCs require mitochondrial respiration to maintain their stem cell function, PCs are mainly glycolytic. Furthermore, mitochondrial metabolism appears to be reduced in the adjacent secretory PCs. This observation implies that during differentiation to PCs, CBCs undergo a metabolic rewiring towards the downregulation of mitochondria. Importantly, there is a lack of impacts on CBC differentiation remains unknown. Mitochondrial morphology is dynamically regulated by cycles of fission and fusion, and this fission-fusion dynamics are key regulators of several cellular processes, such as cell cycle progression and apoptosis. Mitochondrial fission is executed by GTPases, DRP1 and DYN2. The mitochondrial fusion is regulated by GTPases, MFA1, MFN2, and OPA1 that promote fusion. Upon activation, DRP1 binds to adapter proteins located in the mitochondrial outer membrane to initiate the process of fission. DRP1 recruitment is achieved by several effector proteins, including MFF, MID49, MID59, and especially in yeast, FIS1. The fork head box O or the FOXO transcription factors are regulated by insulin or growth factor dependent P13K signaling, glucose, and oxidative stress. FOXOs regulate stemness in several systems, including embryonic, hematopoietic, and neural stem cells. In this paper, they have described the novel function of FOXOs as the regulators of intestinal homeostasis. They show that in organoids as well as in mice, loss of FOXO leads to an increase in the number of secretory cells, that is PCs and the goblet cells, types with a loss of LGR5 plus cycling CBCs. This indicates a role of FOXO and P13K signaling in the regulation of intestinal stem cell fate. Further experimentation and results will be explained by Hemant. Hemant, you can take over.
Okay, thank you, Srishti. Am I audible?
Yes, you are.
So we have total of four experiments, like four experiments, and within four experiments, each experiments have some small experiment to confirm their findings. So at first, they didn't sure about the function of FOXO in intestinal lineages. So what they did was they introduced in small intestinal organoids doxycycline inducible short hairpin construct to knock down FOXO1 and FOXO3 expressions. You guys maybe ask me what is the doxycycline and short hairpin construct. So doxycycline is a tetracycline antibiotic that fights bacteria in the body, and short hairpin construct is a small hairpin like RNA molecule that can be used to silence target gene expression. They confirmed FOXO1 and 3 knockdown by gene expression and protein level after 36 hours of doxycycline treatment. First, they analyzed the effect of FOXO knockdown on the expression of intestinal cells type marker, which includes LGR5, ASCL2, AXIN2, and OIFM. In the graph, you can see on y-axis, we have normal gene expressions, and on x-axis we have different types of cell markers. This qPCR bar graph shows gene expression analysis of intestinal cell lineages marker genes in doxycycline treated FOXO1 and 3 organoids. FOXO knockdown resulted in the downregulation of the stem cell marker. As you can see, those green bars are resulted downregulation of stem cell marker LGR5, ASCL2, AXIN2, and OIFM4. And no significant effect was observed in the luciferase short hairpin control. Intestinal stem cells are proliferative, and in agreement, expression of the proliferation marker Ki67 was reduced, and they observed reduced organoid size upon FOXO knockdown. Decreased proliferation is necessary for cell differentiation. Consequently, they analyzed the expression of several differentiation markers. While they did not find difference in the expression of the enterocytes of endocrine markers, that which one are ALPI, SCT, and Neuro3. They found a significant increase in the expression of the secretory cell markers LYZL, 2, MUC2, and GOF5 upon FOXO knockdown. These results suggest that FOXOs positively regulate stem cell state, and that its downregulation leads to an increased differentiation toward the secretory phenotype. Next slide, Srushti. So RNA expression is not always indicative of protein expression. Therefore, they analyze the protein levels of Punnett cell markers lysozymes by Western blot. And in another diagram, they visualized the number of localization of Punnett cell by confocal microscopy in anti-lysosome and WGA stain organoids. FOXO loss induced an increase in lysosome protein level and in the number of Punnett cells allocated at the bottom of the intestinal crypt. In line with the gene protein expression analysis, they observed an increase in population of Punnett cells upon FOXO knockdown, further supporting the notation of the FOXO knockdown drives Punnett cell differentiations. In order to verify their in vitro observation in vivo, they made use of previously generated LGR5 EGFP villain CRE FOXO13 knockout mouse model. In line with their result, FOXO1 staining in intestinal sections of WT mice showed nuclear localization in stem cells and TA cells and negative staining in Punnett cells and goblet cells. In the given diagram, the red arrowheads indicates Punnett cells, red asterisks indicates CBCs, and green arrowhead indicates goblet cells. and green asterisk indicates transist amplifying, that is TA cells. So the diagram next to that LGR EG5 Valenciari, I don't know the I try to find, but I didn't get what they are telling in the diagram, like showing. So maybe we can discuss at the end of the session. Next slide, Srushti. So Valenciari FOXO 134 knockout mice showed indeed FOXO 1, 3, and 4 knockouts specifically in the epithelial cells of the intestinal lining. Importantly, and in line with the organoid model, knockout of FOXO brings a significant decrease in the LGR5 CBCs and an increase in the secretory cells types of Punnett cells and goblet cells. Interestingly, they found a decrease in the proliferative populations of cells, and their localization seems to be shifted towards transist amplifying compartment, possibly as a consequence of penicillin enrichment in the intestinal crypts. Altogether, both organoids and mouse model supports the notation that FOXO downregulation leads to stem cell differentiation into secretory cells. Next slide, Srushti. So in the second experiment, which is downregulation of FOXO1 3 decrease expression causes mitochondrial fusion through the regulation of MIR484. Their previous work showed the importance of the mitochondrial metabolism in the CBCs. Moreover, they found that compared to CBCs, Punnett cells have lower mitochondrial activity. This indicates that along with the differentiation from CBCs to Punnett cells, a metabolic reprogramming takes place in which mitochondrial number and activity are reduced. FOXO factors are well-known regulators for metabolism. Thus, to investigate the mechanism that leads to Punnett cell differentiation upon FOXO loss, they analyzed the metabolic consequences of FOXO knockdown in intestinal organoids. Bioenergetics analysis showed that FOXO knockdown leads to the decreased basal and maximal mitochondrial respiration and a milder decrease in basal glycolysis. As you guys can shows in the graph, on the y-axis we have OCR, that is uh, basal and maximal oxygen. To further study the consequences of FOXO loss on mitochondrial status, they analyzed the morphology of mitochondria in intestinal organoids. Mitochondria are dynamic organelles that are maintained by cycles of fission and fusion. Fission and fusion rates determine the morphology of mitochondria, from more fragmented to more elongated structures, respectively. Thus, they analyzed mitochondria and their morphology by confocal live imaging and electron microscopy. Confocal live imaging showed a decreased number of mitochondria in Punnett cells when compared to adjacent CBCs. Next slide, Srushti. Additionally, by using electron microscope, they observed morphological differences while stem cells are rich in mitochondria with clearly distinguishable fused and fragmented morphologies. Punnett cells showed less abundant and smaller mitochondria, which is observed both in organoids and in mouth intestinal sections. Next slide, Srushti. Next, they analyze mitochondrial morphology upon FOXO knockdown. They found that while in control organoids, mitochondria displays both morphologies, fused and fragmented. FOXO loss leads to the decreased fused structures and in enrichment in smaller mitochondria. Together, their result shows that FOXO loss leads to the decreased in mitochondrial respiration and increase in mitochondrial fragmentation. Next, they investigated the mechanism by which FOXO drives these changes in mitochondrial status. The balance between mitochondrial fission and fusion is maintained by the activity of a set of dynamic related GTPase that drives these processes. Recruitment of GTPase DRP1 to mitochondria is a critical step in fission. In yeast, phase 1P recruits the DRP1 homologue to mitochondria, and in mouse and humans, its knockdown induces elongation and its overexpression increases fragmentation in a cell type specific manner. FOXO3 can regulate F1S1 levels through MIR484 mediated transcription regulation of FIS1. Therefore, they analyze the effect of FOXO knockdown on MIR484 and FIS mRNA and FIS1 protein. They found that down regulation of FOXO resulted in a decrease in MIR484. As you can see in this graph, the down regulation of MIR484 expression and increase in FIS mRNA and FIS protein levels. Next slide, Srushti. Sorry, previous one. So this suggests that FOXO regulation of FIS1 may proceed through MIR484 regulation and suggest mitochondrial fission as the driver of punished cell differentiation upon FOXO downregulation. To further investigate this, they genetically introduced a doxycycline inducible system for MIR484 expression in the FOXO knockdown. Hence, doxycycline treatment simultaneously induces FOXO knockdown and the overexpression of MIR484. They found that MIR484 overexpression decreases the differentiation into secretory cell induced by FOXO knockdown. Interestingly, the FOXO knockdown induced respiratory phenotype is not significantly restored by overexpression of MIR484. Altogether, these results further support that FOXO knockdown leads to secretory differentiation by activation of mitochondrial fission through MIR484 or FIS1 mechanism. In the third experiment, which is mitochondrial fission required for secretory cell differentiation, they want to assess whether mitochondrial fission drives differentiation into punished cell downstream of FOXO knockdown. They made use of MDIVI1, which is a small molecule inhibitor of mitochondrial fission. They treated FOXO1 and 3 organoids with MDIVI1 and analyzed its effect on mitochondrial morphology by confocal and electron microscopy. Despite the mild effect on mitochondrial morphology by MDIVI1 as a single treatment, they found that MDIVI1 was able to partially restore the increased fragmentation of mitochondrial induced by FOXO loss. Next slide, Srushti. After that, they evaluated the effect of inhibition of mitochondrial fission on secretory cell differentiation. They analyzed the expression differentiation marker and the protein level of punished cell marker lysozyme. Gene expression analysis showed that although MIDIV1 has no significant effect on the expression of stem cell markers upon FOXO loss, it does partially revert the induction of the secretory genes expression markers. In agreement, they found that MDIVI1 treatment could significantly rescue the increase in secretory cells as shown by the penicillin markers and lysozyme and quantification of WJ secretory cells. Interestingly, and in contrast, MDIVI1 treated did not significantly recover the decrease in the mitochondrial respiration upon FOXO knockdown. This suggests that possibly both fission and fusion dynamics and activity of mitochondrial independent of mitochondrial morphology. Recently, a role of MDIVI1 as a complex 1 inhibitor has been proposed, and its role in mitochondrial fission has been revised. To establish whether the effect of MDIVI1 on secretory differentiation could be through complex 1 inhibition, they first analyzed the effect of MDIVI1 treatment of organoids on mitochondrial respiration and mitochondrial redox state. They
Hemant,
did not
are we on
find.
the right slide? Can you just check? I have
Yes, sir.
a
We are on the.
So right now we are seeing a nodal role for Notch FOXO mitochondria axis in stem cell differentiation. Is this where you want to be?
No, sir. Previous one.
This also does not show anything about MD, the small molecule. This is all about DRP1 protein, right? The fission protein. So are we on the right slide?
Sir, yes, sir. We are on the right side. We are
Okay.
getting through by it.
Okay. Yeah. Go ahead. Sorry.
Okay. Okay. To establish whether effect of MDI1 on secretory differentiation could be through complex 1 inhibition, they first analyzed the effect of MDI-VI1 treatment of organoid on mitochondrial respiration and mitochondrial redox state. They did not find an alteration of those parameters upon MIDI1 treatment at the deficit concentration and incubation times. Finally, and in contrast to MDI-VI1, rotenone, a non-complex 1 inhibitor, did not rescue the FOXO knockdown-induced phenotype. In some, Srushti, previous slide, please. In some, their result indicate that mitochondrial fission is required to step in process of secretory differentiation, which is induced by FOXO loss. In order to determine whether direct modulation of mitochondrial dynamics is sufficient to modulate cell differentiation, they manipulated fission by introducing an inducible short hairpin targeting the mRNA of mitochondrial fission protein DRP1. As expected, DRP1 knockdown leads to reduced length and size of mitochondria. Importantly, DRP1 knockdown by itself resulted in a significant decrease in lysozyme protein and in population of secretory cells. Interestingly, and in line with MDIVI1 treatment, induction of mitochondrial fission through DRP1 knockdown by itself does not lead to major changes in bioenergetics in organoids. Altogether, these findings further support the notation that mitochondrial fission is driver event of secretory cell differentiation. Next slide, please, Srushti. So in the fourth experiment, the intestinal stem cell state is maintained by high activity of WNT and Notch signaling pathways and inhibition of Notch driver secretory differentiation. FOXO and Notch have been reported to functionally interact in the regulation of stem cell maintained differentiation in muscles. Thus, they analyzed activation of Notch signaling upon FOXO knockdown. They observed that activated notch intracellular cytoplasmic domain NCID levels decrease upon FOXO knockdown. These result shows that secretory differentiation induced by FOXO knockdown correlates with the inhibition of notch signaling. Organoid cultures can be customized to induce differentiation to specific cell type. In order to analyze the role of FOXO in canonical Punnett differentiation, they grew PC enriched organoid culture by addition of DAPT, which is a notch inhibitor, and CHIR9902, which is an WNT activator. Next slide, Srushti. Interestingly, FOXO level seems to be responsive to notch activity as the induction, no previous one. as the induction of Punnett cell formation by notch inhibition leads to concomitant decrease in FOXO1 protein. Next slide, please. In order to explore this further, they analyzed the consequences of notch inhibition on mitochondria. Similarly to FOXO knockdown, they found that DAPT-mediated notch inhibition induced the levels of FIS1 and mitochondrial fragmentation. Additionally, they observed that while mitochondrial DNA copy number is increased in the stem cell enriched culture, it is decreased in the Punnett cell ones. These results suggest that FOXO and Notch functionally interact and shows that inactivation of both pathways leads to FIS1 upregulation and mitochondrial fragmentation. Next slide, Srushti. No, previous one, please. Okay. Next, they analyzed effect of inhibiting mitochondria fission on Punnett cell differentiation driven by Notch inhibition. Their result shows that in agreement with their result after FOXO knockdown, inhibition of mitochondrial fission by MDIVI1 can partially revert lysozyme induction and the increase in Punnett cell induced by the Notch inhibition. Together, their result show that in the intestine, FOXO and Notch signaling converge to regulate stem cell maintenance, as inhibition of either FOXO or Notch leads to secretory differentiation through a mechanism that require mitochondrial fission and downregulation. Next slide, Srushti. In order to extend and independently confirm their understanding of the FOXO-Notch mitochondrial axis in the regulation of intestinal cell lineages, they analyzed single cell sequencing data from mouse intestine. In four-sided work, performed full-length single sequence RNA of mouse small intestines of 8 mice with total of 1,522 cells in line with clustering of scRNA sequence data by t-distributed stochastic neighbor embedding showed 9 cells cluster corresponding to the main cell lineages in mouse small intestine. Next slide, please. In order to generate a temporal map of intestinal differentiation, they generated diffusion maps in the dimensionally reduced data. They found that the top two diffusion components accommodate the cells in the three branches, while stem cells are located in the center, as you can see is in the graph. Three trajectory branches correspond to enterocytes, tuft cells, a secretory cluster of goblet cells, and Punnett cells on the same branch. Next, they applied the Palantir algorithm to estimate a pseudo time trajectory through the data. Thus, they transform the cluster classification, which is discrete variable, to continuous one that more faithfully represent the process of differentiation. Accordingly, the lowest values of pseudotime were found in center located stem cells, and these values increase along the branch's direction of differentiation. They validated the performance of their pseudo-time estimation by analyzing branch probabilities and gene trajectories of the marker genes of each differentiation cluster. As expected, the expression of stem cell genes decreased along with the differentiation into all three differentiation trajectories. While according to differentiation markers specifically increased within each respective cluster. Of note, the expression of genes that define goblet cells state also increased along pseudotime in Punnett cell cluster and vice versa, suggesting similarly in the trajectory of differentiation of these two cell types. Next, They analyzed expression of FOXO1 and 3 in the diffusion components and along the differentiation trajectories. They found that along with the progression to enterocytes or endoendocrine differentiation, the expression of FOXO1 and 3 does not display major changes. However, it decreases toward differentiation into goblet and punnet and to lesser extent tuft cells. Thus, consist with their experimental results, FOXO1 and 3 are highly expressed in stem state cells and downregulated in the goblet cells and Prenette cell secretory types. As their experiment results showed that the regulation of stem cell maintenance and Prenette cell differentiation through FOXO occurs in a mitochondrial dependent mechanism. They analyzed the expression of genes that encodes for mitochondrial protein as a proxy of mitochondrial abundance. In agreement with their result, they found that mitochondrial markers are high at low pseudo time, closer to the stem cell state, and decreased along a differentiation trajectory into pinnate, goblet, and lesser extent to and GUF cells, while it is maintained along with the enterocyte differentiation. Lastly, next slide. Lastly, they evaluated FOXO1, FOXO3, and Notch1 expression pattern in a cluster independent manner. To do so, they define the expression of the three genes as a unique component to distribute all intestinal cells in a three-dimensional space. Additionally, they introduced a fourth dimension by coloring each cell according to their cluster classification, as you can see in the both the graphs, and alternatively by the expression of level of mitochondrial gene VDAC1. Importantly, this analysis shows that expression of these three genes, FOXO1, 3, and Notch1, is sufficient to segregate the cells according to their lineages. And second, that decreased expression of FOXO1, FOXO3, Notch1, and mitochondria are consistent and robust markers of goblet and punnet differentiation. In line with that, they found that expression of notch transcriptional targets, which one are OIFM4 and HES1, correlate not only with the Notch1 but also with the FOXO expression, especially in the cell types with active notch signaling stem cells and enterocytes. This further supports the functional interaction between FOXO and Notch. Lastly, they perform the same analysis but it on another independent dataset generated by droplet scRNA sequence of 10,396 cells of the small intestine, which led to the same conclusion. Thus, taken together, the independent approach using single scRNA sequence analysis clearly confirmed here in vivo and in vitro observations showing FOXO and Notch signaling converge on mitochondria are key factors in intestinal stem cell maintenance and secretory cells differentiation. Over to you, Srishti.
Okay, so now summarizing everything, the WNT and Notch are the evolutionary conserved signaling pathways that regulate cell patterning during development. Importantly, in the mammalian intestine, these same pathways control stem cell proliferation and differentiation. While WNT and Notch signaling are active in stem cells, in progenitor cells, their activity is polarized. Absorptive progenitors are WNT low, Notch high, whereas secretory progenitors are WNT high and Notch low. In this present study, they have provided evidence that mitochondria play a key role in determining cell fate. They showed that mitochondrial activity supports the CBCs, while adjacent Bennett cells, as we saw in the video, show reduced mitochondrial activity. This implies that CBC differentiation into PCs involves a metabolic transition towards downregulation of mitochondrial dependency. Also, the Fockhead box O or the FOXO transcription factors and not signaling interact in determining CBC fate. In agreement with the organoid data, as mentioned by Hemant, FOXO 134 deletion in mouse intestine induces secretory cell differentiation. Importantly, we show that FOXO and not signaling converge on regulation of mitochondrial fission, which in turn provokes cell differentiation into goblet cells and PCs. Finally, scRNA sequence-based reconstruction of CBC differentiation trajectories supports the role of FOXO, Notch, and mitochondria in secretory differentiation. Together, this points at a new signaling metabolic axis in CBC differentiation and highlights the importance of the mitochondria in determining stem cell fate. Now, the limitations of the study. There are broadly two limitations mentioned in this study, as Hemant has mentioned earlier. One is that it has been recently shown that MDV1, which is a cell permeable selective inhibitor of mitochondrial division, affects the redox state of mitochondria. In this study, using mouse intestinal organoids, researchers did not find MIDV1 altering the mitochondrial redox state. Secondly, the FOXO transcription factors regulate a plethora of cellular functions, so it is unlikely that a single pathway will be responsible for mediating the FOXO-induced regulation of stem cell function. Impairment of the mitochondrial respiration and activation of the P38 upon FOXO-KD are not restored by inhibition of mitochondrial fission, which poses these two factors as potential additional regulators of secretory differentiation that remain to be studied. So that's it on our side. Thank you, and we'll be happy to take questions if any.
Thank you, Srishti and Hemant. Very well choreographed presentation. Very nicely done. I think this is a really beautiful story. I mean, I didn't understand all aspects of their data, but I think it's a very powerful way of looking at molecular data. I mean, initially they started off with simple experiments, and then used microscopy, and then eventually independently did single cell sequencing. So I really liked it. I have a bunch of questions. Of course, you also alluded to at least one slide where things didn't make sense. So what we can do is maybe we'll open up the questions to the whole floor, and but we can go ahead and start with that one particular slide, Hemant, you showed, which you said it didn't make sense.
Yes.
So maybe you can ask it by yourself, like, okay, what was not obvious, and maybe somebody is able to pitch in, and if they understood, they can at least start discussing. So if you can show that particular slide, Srishti.
Sure, sir, I'll share it.
So there are a couple of technical things that I am actually reasonably well aware of. So I will wait for someone to ask question about that particular thing. If not, I'll bring it up. So I think a slide. I didn't note the slide number, but keep going.
Sure.
I think that's where they had the red box, red triangle, green triangle, red asterisk, green asterisk. The microscopy.
14th one, I guess.
Before that.
14th one.
Okay.
This one.
Cool. Okay.
No. Yeah.
This is where I thought you.
Went back. Just a minute. Yes.
I think, Hemant, you stopped on this slide and you said you didn't quite understand this. Am I right? Or was it a different slide?
Sir, I understand the LGR5 EGF and to the both image, I didn't understand in CRE FOXO 134 knock.
So why don't you explain what you understood here? So what exactly? So now forget about the data. Forget about the data. Can you explain? And anybody can chime in if they want to. How did they? Forget about the result. Just forget about the result. What is a question, right? Yes, only in this slide. What is the question that they're asking? How did they design the experiment? You can take a minute to think about it. So what is the question that they're trying to
Sir,
address?
I think
Go ahead. Yeah, go ahead.
I think they try to verify in vitro and in vivo observation, so that they take that samples and shows that
Specifically, what is the question?
that markers to verify in vitro and in vivo observations, I guess.
What of what? So when you look at this, I'm looking at, I don't know anything else, right? So just by looking at the words FOXO 134KO, it's very, and you mentioned it there on the top. Basically, they want to knock out all the FOXO genes, right? They want to knock out all FOXO genes. And then
Yeah.
ask, what is the consequence of knocking out all three FOXO genes in the intestinal crypts, if I'm not mistaken, right?
Yeah.
So this is a mouse model. So how do you create a mouse model? How do you make sure that FOXOs are deleted? Specifically see, you already mentioned early on that FOXO proteins are important for so many different things, correct?
Yeah.
So what if you remove FOXO in the entire body? What will happen to the animal?
Many processes going to stop, like means pathways and all.
Okay, then will you be able to find out its role specifically in the cells you are interested in?
Yes.
No, if the whole body is reacting to the loss of FOXO proteins, can you tell what is happening specifically uh, in the intestinal cells?
It will be difficult to be specific, and
Exactly.
that's why we
Yeah.
target, we knock down the FOXO in that particular process.
How do you specifically knock out these genes only in the intestinal cells?
That's what they said, no, by introducing
By
a hairpin
organoid.
structure.
No, that
Yeah.
is in the embryoid. That's in the organoids.
Yeah.
This is an animal model, right? This is an animal model. So in
Right,
the
right.
animal model, how do you remove a particular gene or a set of genes specifically in only one tissue or one cell type? The clue is already on the screen, but I just want you to think. By the way, the question is not only for Srishti and Hemant. Anybody who got the clue can probably talk about it. Is the question clear? How do you remove FOXO 134 only in the intestinal cells?
Yes, sir.
So they're saying that it's in vitro observations.
So the previous slide, if you go to the previous slide,
Sure.
that is the in vitro. So the nice thing about this particular story is that they have two. This is an in vitro, very beautiful in vitro study where they are growing, uh, organoids in culture. Now, why did they take organoids instead of normal cell culture? Normal cell culture, you can have only one cell type, but you want a system where there are multiple cell types, correct? So therefore, you used organoids.
Yeah.
Now, organoids are growing in a tissue culture flask. in the organoids, how do you get it?
Yeah.
And you take it from normal cells probably, and then because you want them to work normally initially, and then you want to knock out or you want to remove the gene function. So when a group of cells are growing in tissue in a tissue culture flask, how do you get rid of gene function? By the way, it's a very simple technical question. I don't want you to think about very fancy things right now, because most of these experiments that they did, which I think very elegantly they showed, right? Their laboratory has two strengths. They are able to work with embryoid bodies, and they're also able to make animal models, right? So this is what will happen to you if you are in a laboratory. You're not trying to answer very big questions about all. So you're saying, okay, how do I get rid of FOXO 1 and 3 in my organoids? If you forget about this slide, if you take 30 seconds to think about an experiment, how do you do that? How do you knock out a specific gene in tissue culture? Take 30 seconds and anybody can chime in. Is the question clear? Anyone?
Yes, sir.
Okay, good. So we'll take 30 seconds and then come up with an answer. Okay, I should ask, is the question not clear to anybody?
Me, sir.
Okay, good. So you are interested, sir. Good. Pragna also is asking. The question is, we want to find out FOXO protein's involvement in whatever, doesn't matter what. Okay, right now in this particular case, they want to find out what happens to certain cells. Now it turns out in a complex system like the intestinal crypts, where there are multiple cell types, right? In the introduction, it was very clear that there are multiple cell types in the intestinal crypts, correct?
Yes, sir.
Good. Now, if you only take one cell type and study in the tissue culture, which is what many people do, you will lose some information, correct? You will not know what is happening in terms of their interactions, and it's not really the real life situation. So the next best thing, which is not easy actually, they are doing a lot of now. It's one of those big things they are doing these days, is they are able to figure out how to grow organoids from the intestinal cells, and that itself is a big feat, which is fantastic. Now, again, imagine yourself in the laboratory. I think most of you have done the Labster modules, right? So if you imagine going into the laboratory and going to the tissue culture, so you brought out a flask of organoids. They're normal looking, healthy. How do you get rid of specific genes or gene function in those cells, in the organoids?
Sir, I think that we already know that there are like naturally occurring mutations happening which can knock down these genes. So what if we identify them and then inactivate by establishing like DNA sequencing or other such methods?
Can somebody be a little more specific?
Uh,
Go ahead.
sir, I think we studied about endonucleases. They specifically cut, I think, DNA at specific points. No, maybe. Restriction endonucleases. Can they be used?
Very good thinking. So what you are in the right direction, but so we already talked about CRISPR, right? You could potentially use CRISPR to knock out these specific genes. Now the problem is it's not trivial to get CRISPR into the. I mean, if CRISPR works, that's fantastic. So what Srishti mentioned about, Srishti was saying, maybe we'll identify naturally existing mutations in FOXO. Now, on this planet, where do you look for them? Naturally existing ones, especially organoids. One possible way is to find patients who already have
Cancer
mutations,
patients, yeah.
right? Potentially, you can find patients, and that's what will direct you. Then,
Right.
if you can get a biopsy of their intestinal cells, not very easy, but possible.
Yeah.
Then you can grow in the culture. Now, it turns out it's not that trivial to get. I mean, if you want to get all FOXO3 protein mutations in a patient, the patient doesn't exist because the person might not even live. I don't know, right? So you are limited. You cannot. It would be fantastic if you already had cell types which have mutations, but you don't have that. So you grow normal cells and make organoids. Then Tejaswini said, "Oh, I will use enzymes, endonucleases." Now it would be fantastic if we are able to deliver that, because we can. The problem then would be, how do I know that they all have lost the gene? Because you have to test it, right? Even if some cells are still having expression, I mean the gene is intact, you will still have. Now that is still a problem. So this is the obvious thing on the screen itself. You see the SH Luke and SH Foxo13. SH stands for short hairpin. That is essentially RNA interference. So they use DNA constructs which make short hairpin structures which work through RNA interference. RNA interference works at the mRNA level, and unlike CRISPR, which works at the DNA level, the RNAi. By the way, about around 2000 to 2003, and one set of Nobel prizes were also given to Greg Hannon and I forget Craig Mello. Craig Mello and Greg Hannon got a Nobel Prize on RNA interference. Now, there was a time 10 years ago, I would only talk about RNAi. The way I talk about CRISPR now, there was a time I would only talk about RNAi, because that was the best thing at that point. It was breakthrough technology. So, but even now, a lot of people do RNA interference based knockdown of gene expression. Okay, so the Luke is for luciferase, to show that luciferase is not an endogenous gene, so it's a control experiment. So when you knock down luciferase, they are saying, okay, they look at the band intensity. So how are they actually making sure that the short hairpin structure is expressed? So it's a DNA vector which has a capacity to produce single hairpin structures, RNA structures, which can bind to mRNA and degrade them. So instead of creating mutations in the DNA, you are attacking the mRNA. And is that clear? So Alekhya, because you asked a question, I'm going to use you as my feedback. I'm addressing everybody, but I need somebody to at least kind of acknowledge that I'm making sense. Anybody can, but are you with me, or have I already lost you?
No, sir. I think I understood what SH Luke is used.
As a control.
Yes.
And when you use, keep yourself unmuted. I'll ask you a couple of questions and then we'll move to somebody else. I don't want to only point to. So when you use control shRNA against luciferase, what do you see? So, and now there's another addition of doxycycline. There's a minus for doxycycline and plus for doxycycline. Why do you think they used that? What is the purpose of that minus plus? You can think for about 10 seconds. Any thoughts? What could it be? You might be wrong, but you want to guess?
Some kind of
Maybe DNA have negative charge.
No. That plus minus.
Is it related to, sir, addition or deletion of
or some
the
kind
genes?
of staining, maybe?
Okay, I think who said that earlier? Nidhi or not Nidhi. Somebody else said about addition or removal of something.
Yes, sir.
And Alekhya, you said?
I didn't say anything, sir.
Okay, sorry. So the plus minus is addition or no addition of doxycycline. And Hemant already mentioned that doxycycline is an antibiotic. The advantage of doxycycline here is, now they don't talk about it in specific because it's a very generally used technique in the laboratory. Doxycycline, okay, you all know about this. Hopefully some of you know about this. You are trying to clone a gene and put it into bacteria. Okay, you want only those cells which have the DNA of your interest to grow. Can you make sure that, number one, all cells have the DNA of your interest, or only cells that have your DNA survive? Which of these two things is possible and you can make it happen?
I think cells that only have the DNA.
Second one seems possible.
Yeah.
Why and how would you do it? What is the problem in expecting that all cells will get the piece of DNA that you're interested in? What is the bottleneck there? If there are a million cells, can I experimentally be assured that all cells will get a piece of DNA that I'm interested in?
No.
It's hard. You can't predict. I mean, yeah, go ahead. Anybody else? Okay. So Alekhya says no. So maybe we have to devise a method where only those cells which take up my piece of DNA survive. Right? So both Srishti and Alekhya kind of alluded to that. How do you make that happen? How do you make sure that only those cells which take up your piece of DNA in a transformation experiment will survive? You kind of have an idea, at least by reading or in textbook stuff. I think. I might be wrong. So that's where you have to think slightly. You have to get out of the DNA and ask, where is it growing? Right? It's growing inside a bacterial cell. So how do you make sure that only some cells, some bacterial cells live and other cells die? What technology can you use so that only some cells, so some bacterial cells survive? That cell, those cells which have taken up your DNA.
In that, sir, am I audible?
You are.
Sir, in that gene of interest, we can add one more sequence that will make one antibiotic for same kind of species of bacteria. So when our gene of interest get translated, that compound will also get translated and that will kill other cells. We can do like that.
You are getting there. It's difficult because if that compound will kill other bacterial cells, what is stopping it from killing itself, right? But I think what they do is you essentially try to kill all cells by using an antibiotic. That much you know, correct? But now, along with the piece of your DNA, you put an antibiotic resistance gene. So that's a selection. You select your cells because as long as your DNA, as long as the antibiotic resistance gene is there. Just to give you an idea, this is an important thing that you will need to know because it's actually very fascinating. There are genes. So while antibiotics are made by bacteria and fungi, bacteria themselves have antibiotic resistance genes. For example, there are a lot of beta-lactamase enzymes. Not beta. Antibiotics like penicillin, right? So, penicillin, I'm sure some of you know. How does penicillin work as an antibiotic?
It stops the cell wall formation.
How? So this is again. I am almost modeling what you should do when something is being told to you. I mean, then you should ask why. Maybe we don't know the answer, but that's an important question, right? So it turns out penicillin has a similar structure like the beta-lactam rings in the peptidoglycan thing. I'm not even an expert in this. I'm sure these guys, we had a discussion in St. Anne's about this a little bit, not too much. And they they were able to tell me a lot more about the peptidoglycan structure. Anyway, so penicillin goes and sits along with the peptidoglycan, and now the peptidoglycan cannot extend during the cell wall biosynthesis. So you're absolutely right, Chaitanya, right? I forget who's talked right.
Yes, sir. Yes.
Okay. But there are beta lactamase enzymes. Those can cleave penicillin-like molecules. They can actually destroy them. So, so it's a protein. The protein is encoded by a gene sequence. So if you put the gene sequence along with the piece of your gene interest, your gene of interest, as long as the beta lactamase is being expressed, your DNA of interest also is being expressed. That's how you select it. Now, the reason I mentioned this is about using biochemicals for selection. Now, in this particular case, doxycycline is used to preferentially, so you create a construct like you create an antibiotic in one situation, antibiotic resistance gene. Here, you create doxycycline has the ability to bind to certain, essentially it acts as an inducer of gene expression. So there are, I think doxycycline either binds to certain proteins or can modulate transcription. So in order to make sure that only you have control over gene expression, you use doxycycline inducible short hairpin RNA molecules. So doxycycline will induce the short hairpin. Short hairpin will go and bind to mRNA of that particular gene sequence you are interested in knocking down, right? I hope we are getting to some place. I've taken too much time, but I thought it was an important thing to, when we show this particular graph, this figure, it's very important to figure out we should be able to recreate the experiment in our minds. Okay, so in the interest of time, I'll quickly go through. I mean, you should really do this carefully, not necessarily for the presentation. So we have certain goals for doing the presentation. We want to go through the whole study, but you should take up at least one experiment and literally deconstruct every part of it. Just by looking at, you can either read the materials and methods, which is fine, but just by the way you'll get better at this is when you look at an image and you will tell what it is. For example, you go to a restaurant, you can look at the menu, or you can go directly, and if it's a buffet, you can just go and look at it, right? What will you do? Would you read the menu and imagine, or would you go directly to the stalls where they put all the food and decide what you want to eat? Which one would you do?
To the stalls.
You go there, you say which is fresh, what is nice, what is appealing, what other things. By reading the menu, I mean, it's a sidetrack, our usual tangents. But by looking at the figure, like looking at the stall where all the food is there, you now try to figure out how it's made. I'm sure if you are a cooking enthusiast, as soon as you go to any food thing, you will start thinking, right, how did they make it? And that's the same attitude, by the way. It's not very different. So as soon as you look at this, you are trying to deconstruct this entire thing and say, how did they do the experiment? So this seems to be anti-lyse. This is actually staining. We'll come to that later. The right side. The left side is, the good thing about this slide is it's actually mentioned everything. Therefore, they analyze the protein levels of Paneth cell marker lysozyme. So lysozyme, that's in fact, just by looking at this particular data, I'm learning that lysozyme is specific in Paneth cells. Now, how did they get Paneth cells only? I don't know. But maybe Paneth cells are the only one in the intestinal cells, or the organoids, by the way, I think. Probably organoids. Yes, this is an organoid experiment, so my bad. So if you crush the embryoid organoid, you will find lysozyme, and lysozyme, they probably showed already that it's only in the Paneth cells, probably. That's why. So you see the intensity of lysozyme in the combination. Look at this. SH-FOXO13 and doxy plus is the only place where the intensity of lyse is more. Would you agree?
Yes, sir.
Okay. Now, how are they not cheating you? You'll ask, wait a minute, how do I believe you? If they only showed this one, right? If they only showed the right extreme one, you don't know what you're comparing it against. So you are saying, wait a minute, I want to convince you that only when I knock out FOXO13 will I see an increase in lysozyme. How do I do that? I have a short hairpin struck RNA which targets FOXO13, but it is not expressed until I add doxycycline. So there I have a negative control, that is no doxycycline. When there is no doxycycline, I don't see an increase in lysozyme. But when I have doxycycline, I see an overexpression of lysozyme. Now, if I only showed you the top panel and only those two, just imagine I only showed you lys, doxy plus minus, and shRNA for FOXO13. I say, wait a minute, I don't know, maybe you are loading double the or 3 times the amount on the right panel, on the right column, and you are fooling me. Then I say, okay, if you don't want to believe me, I will do a tubulin. Tubulin is a positive control here, also called a loading control. So in the same lysate, I crush the organoids and I load it. I will look for lysozyme, but I will also look for tubulin, just to convince myself and you that I'm not loading more. Because in an experiment, how do you know, right? You need to have a reference. So are you all with me? Somebody, is it making sense?
Yes, sir.
Okay, one more person so that I.
So I have a doubt.
Yes, please go ahead.
I might have gotten a bit confused. So basically, doxycycline, which is
Doxycycline.
there. Sorry, doxycycline, that is used to knock down a particular.
No. It is used as an inducer of gene expression.
Okay, and over here the plus and minus is plus is where it is present,
Added.
and minus is where it is not added to the short hair.
No. So the DNA. So it's kind of complicated, but I mean, you'll figure it out. It's not very difficult. It's just a little complex. So you want to just imagine doxycycline as a switch. Imagine doxycycline as a molecular switch. Only when you switch it on will the shRNA be expressed. If you don't add doxy, the switch is off, and no short hairpin RNA. No short hairpin RNA, no protein reduction. Are you with me?
Yes, sir.
Okay.
Yes, sir, got it.
Okay. Now the idea is we'll finish up this fast, and then maybe we'll go on to the next thing briefly. I mean, and I thought a couple of things are very important here, and I also want to talk about the mouse model after this very quickly. So now I show only the 2 lanes. I say, okay, fine. I convinced you that only upon adding doxycycline I will actually express the short hairpin RNA. Short hairpin RNA will attack FOXO13 mRNA. FOXO13 mRNA mRNA is destroyed. So no FOXO13 protein, and then lysozyme. So there's a lot of inference, you see. There's no direct evidence. It's all indirect evidence. So I'm showing, okay, if I remove FOXO13 protein by using short hairpin RNA, which will destroy the mRNA, then lysozyme increases. And my loading control is normal. So I can believe that this intensity, increased intensity for lysozyme is real. Now somebody might say, well, how do you know it's actually not? Anytime you put doxycycline, it doesn't matter whether it's FOXO or any other gene or any other random short hairpin, you will still get lysozyme to go up. Then I have to show you, in fact, just to convince myself, I need to show that it's not happening. So I take luciferase, which is not present in the organoids. It's a luciferase gene that, firefly. Luciferase is an enzyme important for creating that light in the fireflies. It's a beautiful biology, and I don't know if anybody got a Nobel Prize for that. But anyway, so I'm using that as a control and say, look, when I target luciferase, which is a random thing, I don't see an increase in lysozyme, but only when I target FOXO13 will I see an increase in lysozyme. Does it help? Is it better than before?
Yes, sir.
Okay. So the right side, I don't want to spend too much time. The right side, they like, instead of the Western blot will tell you a big, an indirect whole picture, but you don't know what is happening in specific cells. So they did instead of antibiotic and antibody labeling, they use something called wheat germ agglutinin is called a lectin. Lectins are proteins that bind to sugars. Now there are lots of people who have studied these things. Essentially, they figured out that WGA binds to certain cells. So WGA is for probably the all of intestinal cells, and anti-lysozyme antibodies specifically for the Pennant cells, which are in the. So you see the DAPI is for all nuclei. Lysozyme LYS is an antibody against lysozyme. WGA is which will bind to Pennant cells, and WGA is wheat germ agglutinin, which binds to all intestinal cells. This is an inference. I don't know. If I read the article closely, I might learn a little more. But here they're making a point that lysozyme is very specific. Only upon introduction of control shRNA, not much is happening here. But I don't know what their point is here. But upon adding doxycycline, FOXO13 is gone, and I am changing the structure. So basically, what used to be only 3, I'm getting more cells here, is probably what they're pointing out. So you see that yellow asterisk marks, they are reduced, they are increased when doxycycline is added. And then lysozyme, when they're looking for lysozyme, that number is increasing as well. So they are making some comment about it. But the idea I wanted to spend a little bit of time on this is, on the left hand side using a Western blot analysis, and on the right hand side using antibody / lectin staining and confocal microscopy, they are trying to talk about the consequence at the protein level and at the cellular level, what's happening. Hopefully that was useful. I mean, I know we are not progressing in the paper itself. So the next slide is about the animal model. This will probably take me another 20 minutes, so I will not talk about it right now. We can talk about it another time, by the way. But something to now, how do you do that in animals is a question. That's the only thing is, you can do that by adding this whole doxycycline, all that stuff in cell culture. But how do you make sure in an animal you can get rid of certain genes only in those cell types? And the clue is this Villen-Cree, Villin Cree. It's the beauty is Cree is from a bacteriophage, and it's a recombinase. And what does a recombinase do? And this has actually got something to do with something related to transposable elements, kind of, because transposable elements are also like recombinases. So, and Villen is a promoter. So there are lots of hidden things inside this particular image. And the reason I kind of spent all this time is for you to, while there are 2 ways of reading a paper, right? You are just going through and you're making sense of one layer. There are multiple layers in any paper. And today's presentation clearly showed, by practice and the general understanding, comprehension of the paper, I think it was very well done, personally. The top layer of this story, which is, I'm talking about, that I think over the last few months, almost everybody got this. You are able to go through that top, and I will hear from all of you. I think you also feel that difference, right? You are able to go through that top layer of going through a paper a little more efficiently than before. What I'm trying to do now is, I want you to go to the deeper layers and ask, okay, I'm able to talk about this. I'm able to talk about this. Okay, knockout, when knockout happens, this happens. I'm able to generally construct an argument based on the data that they are trying to show. Correct? You're able to say, okay, what are they representing on x-axis? What are they representing on y-axis? At least the graphs you're able to handle much better because it's numbers. But when it comes to some of this data, microscopy and other things, and genetics and molecular biology, you should start thinking a little more in those, like statistical analysis. You should ask, what is In graphs, it's easier, right? You ask what is on the x-axis, what is on the y-axis. Similarly, you should ask, okay, what happens here? Okay, it's a microscopy image. It is from this, it is this. Instead of x and y-axis only, there are many more axes here. What is the genetic background? How is it being activated? What is the marker cell that they're doing? What is the technology they're using to look at the marked cells? Those kinds of things. So I will leave it there, and let's do the usual. I know it's already beyond our 1.5 hour mark. So well done. I think it's a very well rehearsed thing. It looks like you stole very nice all the good things from other presentations and you're incorporated effectively. So good job on that. So couple of suggestions. You've written a good script, and it's very obvious. I was able to tell that you understood the paper when you went through the whole thing. The introduction was fantastic. It gave a good overview before you jumped into some of the data, but that connection was sometimes lacking. Now I understood it. I actually was able to follow the whole talk, and I thought the introduction helped me analyze the rest of the talk also properly. But you can make the connection slightly more, meaning when you
Yes.
come back to some of the data slides, you can always say, as you might remember from the introduction, and you repeat something. I already told you blah blah blah, and you take it. So there you are connecting your audience back to the introduction, and you're reinforcing. That is one thing. Second, again, I said it last time also, I'll say it again. I think the scripts are getting better, but you should get much more comfortable with not just the script, but it should flow naturally. So even if you're stopped, you should be able to, I mean, scripts always help. I think even for me, sometimes I do my best when I write my script, but you should know your script in and out, like completely, which will only happen with practice. But that I just wanted to put it out there. That is something you guys can work on. So I will stop there,
Yes, sir.
and
Sure,
I
sir.
will let's do our round of one second, one round of I'll call out, and you guys can tell me what you liked, what you learned conceptually, biology from a biology standpoint. What did you learn from a presentation point? What did you like, and what you might steal, and any suggestions you have. So Nikita.
Sir, can I do it later? My net is fluctuating
Sorry,
a
no
bit.
problem. No problem.
Yeah.
So, first person, let's take a volunteer, and then I'll start assigning. Let's take a volunteer who's ready to go, so that the second person will have some time.
Okay, sir, let me start.
Please go ahead, Chaitanya.
It was well structured, and also, as sir said, it was well rehearsed, and also the alignments of the content they made was also good. And from biological concepts, I learned about foxos and their importance, and also the interdisciplinary importance, such as stem cells and other diseases. And I think it would be better if the background was light, like they used dark background. So that are my views.
Thank you, Chaitanya. Nashra, are you ready?
Hello, am I audible?
You are. Go ahead.
Okay. Yes, so today's presentation, like if you talk about the presentation wise, it looked really attracting, and it was, it was like a thing which attracts you to listen throughout the presentation. So the presentation was really nice, and initially it was going really well, but I was a bit lost in today's presentation, so I'm not at a point that I can comment on their like how did they win, but I think it's my fault because I was lost. The I was not able to connect the dots, but
Nashra, it's
overall,
not at all your fault.
no, but I think it is because I tried to understand, but somewhere in the middle I was missing points. So I'll read the paper and will let you know again. But overall, I think the flow was nice. And at last, when the discussion was going on, I really enjoyed it.
So Nashra, actually, both for you and Srishti, you are right. I think the whole exercise, I know we are not religiously following this, but the most benefit will happen when you also read the paper, right? So you might not have all the details, but if you also read the paper, you'll get more of it. So absolutely, you're right, Nashra. But talking in favor of your comment, the fact that you got lost and I got lost too, and I'll tell you why. That was a case where both Hemant and Srishti were very comfortable. They knew I mean, at least from my understanding, they know the story. They were able to nicely navigate, but they lost. I feel they lost the fact that they lost that connection that they are telling a story to us. This became so comfortable in a journey. Again, that's one way of doing it, right? Like a
Yes,
poet,
I felt it at one point when
Hemant.
Hemant was describing about those figures. I felt that they are so much into themselves that they
Correct.
forget that we are here, and they want to, like, they have to tell it to us.
Now,
So
that is actually true for all of us, by the way. It's actually true with every presentation. Somewhere in midway, you are so comfortable with the flow, and you know, okay, it's working well. It's nicely. You lost them. It's almost like imagine a tour guide. Some tour guide took us to France, all of us, and that person loves France. Let's say we are standing in Paris, right in front of Eiffel Tower, and he's already talking, and we already understood a few things, and then he moves on. He walks away, and he's on in his own world. He's enjoying Paris so much himself that he forgot that he's actually doing the tour for us. You know what I'm saying? Similarly, it's
Yes,
very
sir.
natural. It's a very natural thing. It's a very natural thing for all of us, and it is not just you. You can imagine how bad things can become when a scientist is talking about their story. You can
Yeah.
imagine they are so much in love with their story that at some point they don't care about you. They are so happy about their own work. And I'm not just making this up because oftentimes a lot of scientists don't make sense if they're talking to uh, non-scientists because they forgot how to be tour guides. And that is why these days a lot of this emphasis on science communication, public outreach, because scientists are not being trained on how to reach out. Now a lot of times within scientific, I mean, our conversation happening right now is not very different. If when you go to even professional meetings, it's not everybody's expertise, right? Intestinal villi, all this stuff is not everybody's interest or expertise. So initially, like, I think on an average people are generally connected to the talk for about 5-6 minutes, and after that depends on everybody's interest, but most people fleet away. In a conference there is be like you'll be waiting for your next talk, so there is no time to look at and analyze each talk. Now this kind of conversations and this dissection happen uh, in usually in graduate schools, in journal clubs and other kind of, so that's what we are kind of emulating. So it's not surprising, but it is a responsibility of the speaker to keep connected to the audience and understand that. And it is not easy. I'm just saying that it takes practice, and for everyone, even including me. So Chaitanya is done, Nashra is done. So I'm not going to put them in
Sir,
spot right now. Yeah, go ahead.
I
No,
no
Nikita
no
is
no.
there.
Yeah.
Nikita is ready. Go ahead.
Yes, sir. I mean, first of all, Shrishti and Hemant, they actually prepared us for reading the paper in Kahoot challenges and all. And while going, it went very smooth, but yeah, I got confused in the middle. And then when sir started the discussion, I thought I was getting on track, but then again I missed it. I felt that I should read the paper thoroughly and then comment on you guys. But overall, it was very good presentation. Thank you.
Thanks, Nikita. I was kind of busy the last few days, so I didn't participate in some of this. I read a couple of those articles that Hemant put, but I didn't do the Kahoot. I'm looking forward to doing the Kahoot. So, and Noreen says the Kahoot helped me gain insights about the paper. I also lost several times in the middle due to bad network. I know that's why Noreen has been silent today. But the fact that they understood the paper and presented it is really good. So, Noreen, I would assume that is your comment for the day. If not, I mean, I'll call out your name, and if you send a message, I will not ask you to talk because of a network. Otherwise, I will call out on you also. Not to put you on the spot, Nupur and Samyukta, and I think we lost Jahnavi. So at the end, I would like to get your reflections as well. So just getting you ready for that. I'll ask you the last. So Alekhya, do you want to share your thoughts?
Yes, sir. So I think Srishti and Hemant did a really good job because of the challenge. Actually, I think I read 3 to 4 parts of the paper, and maybe that was why I was very interested in today's presentation. And I think it was during Hemant's part I couldn't understand a few diagrams and all, and I think it was maybe my fault. I really couldn't get into the terminology, or I lost connection with the topics or how it is related to the diagram. But overall, I think they did a great job in understanding such a tough paper from my perspective. So yeah, great job, guys.
So what did you learn from a biology standpoint, and what would you steal from today's presentation?
I've learned a lot of new things. Their topic, I think it was during the briefing presentation also. Foxo was something new to me. I've never heard it before, like ever. So today again, that, and then about those notch, and then how these SH ones work. And there are a lot of new things. I was actually going through the paper while listening to them also, because I was trying to understand something somewhere. I know it was really interesting, but very, very interesting.
Thank you. Palak?
Yes, sir. Uh, uh, Talking about the Kahoot challenges and all they were posting in the group, sir. I was not able to participate very well, so I had no idea about the paper. But in the presentation, sir, I liked the slides and the presentation was very well structured. But about the topics, I think I need to read the paper once again to understand all the single cell, the diagrams that they were explaining about stem cells and everything. So it was a very good presentation. And about the learning part, I think I have to read the paper once again.
Thanks, Palak.
So,
So just a couple of small things. One is in scientific papers, and it's not wrong. It's just what we use in the scientific community. We don't call them diagrams. These are figures. So these are figures and figure legends, and so use the word figures because that's what people are used to. The second thing,
yes, sir.
it's a minor point. That is, it's WNT is actually we pronounce it as winds, wind signaling and notch signaling, and you'll see a lot of these. These are these are standard. By the time anybody finishes a PhD, they're talking about at least 5 or 6 of these signaling pathways: notch signaling, wind signaling, TGF beta signaling, all kinds of NCAM signaling. So it's almost like saying, Hyderabad, Nagpur. When somebody doesn't know anything about these places, they're just names. But somebody who's lived in Hyderabad, I mean, just imagine, you'll say, "Oh, I lived in Dubai." When you say, "I lived in Dubai," suddenly it's not just a name, but you have an experience. So similarly, when you say wind signaling, scientists get really thrilled because they are not just thinking about a word, but they know this whole pathway, all the players involved, and how they interact, and what are the consequences, and stuff like that. So that's how people talk, and it's a matter of getting familiar, right? The more you live in these signaling communities, you'll understand better. Tejaswini. Okay, Tejaswini, if you can hear.
Yes, sir. Am
Go
I
ahead.
audible?
You are. Please go ahead.
Yeah. Yes, sir. So talking about today's presentation, I think Srishti and Hemant did a very great job because of the Kahoot challenges, I went through the paper, not completely, but yes, the abstract and the introduction helped me a lot to understand today's presentation, and it got interesting for me. Uh, and So I was pretty excited for today's presentation, and the way they presented it, the narration and they structurized the presentation very well, I would say. I um, During I think Hemant's part, I was a bit confused with the figures and all, and also I had network issues, so I had to reconnect myself again. But yes, since the terms were familiar to me, I was able to understand what they were trying to convey to us. Uh, uh, and talking about what I learned conceptually today, I think Foxo was a very new thing, and so yes, I learned about it, and also a different kind of signaling pathways, as you mentioned, and also when we were discussing about the doxocycline. It also actually when you discussed about it, so I got to know. I was very um, I understood it very well, and also in a deeper perspective, I would say. Thank you.
Thanks, Tejaswini. Nidhi.
Yes, sir. Am I audible?
Yeah, your audio is actually really bad right now. So I'll come back to you. Check if you can make it better. I'll come back to you. Rachna?
Okay.
Okay, Rachna might be busy. Pragya?
Okay, am I audible?
You are. Please go ahead.
Okay, so their presentation was very well organized, I must say, and as Nashra said, it was very attractive. But I kind of agree with Chaitanya that maybe they could have used a light background, and I learned a few things, not a lot, because I was getting disconnected again and again. I was having some network issues. I got disconnected at least 3 to 4 times. So I learned a few things, and I think that some terms could be explained properly, and some terms got clear while the discussion after the presentation. So and as you said, so that when the speaker is speaking, like they are presenting, they kind of lost connection. So I felt that too, like Hemant was speaking for a very long time. So I think that this maybe they could have just changed the like two experiments explained by Hemant and two by Srishi, like that something. So maybe that would not be a problem. So overall, they did a good job, I think. Thank you.
Thanks, Pragya. So the point is also that, right? So it's this is our platform where we can be mindful of these traps that everybody gets into. If you can go back to your own presentations, I'm sure you are like so into your presentation that you are more interested in getting through your presentation next one. You know what's coming up next, and it only gets better for you. Like, okay, this slide went well. Now to the next one. Now to the next. You've rehearsed. Now there is a certain pace that you want to follow. You are not very comfortable. Somebody is stopping you, which is not very natural. So that from that point of view, if you are trying to be forgiving of the speaker, they want to take you through a story. It's almost like if we fall asleep in a movie, which we don't, and the director is, we are trying to reach a point from a presenter standpoint. We should become like the best movie possible, and from the audience, it's the engagement. I slept through some movies where I was dragged into. I just didn't want to go, but somebody dragged me to the movie, and I would go and sleep. Then if they wake me up and say, okay, did you like the movie? I'm like, okay, whatever it is, I don't know, and I'll probably remember a few things here and there. So the reason I'm saying that is, if as I think Nashra also mentioned, a couple of other people also mentioned, if you read the paper a little more, I think things will get better. So, Nidhi, are you in a better connection?
Sir, now
Yeah, bad audio again. So sorry. So maybe if you don't mind, I know because the audio is very bad. Can you say
okay,
one?
sir, can I leave a message?
Yeah, the message would be really helpful, and I'll read it out.
One.
So
Okay.
while Nidhi
Okay,
is
sure, sir. I'm sending a message.
thanks, Nidhi. Hopefully we'll have a better connection. So I'm waiting for your message that I'll read. But while Nidhi is typing her comment, Srishti and Hemant, can you reflect on your journey the last couple of weeks or whatever you've been doing?
Yes, sir. Shall I go ahead?
Um, okay, so 1 second. Yeah, please go ahead.
Yeah. So, this presentation specifically added a lot of value to my knowledge, because not just because of the scientific part, because of today's comments, because like I got to learn a lot of things, and while I understood what they were saying, I knew exactly what we did wrong. So this is a very interesting thing, because we can catch that up for our next presentations. And the process was amazing. Just like you said, we were so into the journey, we were so into the paper, the story, and the presentation, we forgot that we have an audience to address. And that is again a very important and valuable thing that I got to know today. So yeah, learned a lot of things and enjoyed everything a lot. Thank you.
Hemant?
Yes, sir. Sir, we prepared a lot for this presentation, like I think, from the point where you gave us the paper. And I think I know, like, in my part, I can do better. I just like went on, means it's like a train, it's nothing like, means ask anything or anyone to, like, if they understand or they are getting their point. I can improve in my future presentation those things. And the whole process of going through Foxo and this paper and the review article, I really like the Foxo and want to know more about the Foxo protein. So yeah, this is amazing journey, sir.